Biochemical Engineering and Enzyme Discovery. Introduction to the module. Biochemical engineering is a branch of science that focuses on the performance and adaptation of known biological processes on an industrial scale. The field combines the fundamental principles of biological sciences along with that of chemical engineering. This module examines the uses and concepts of this fascinating field of research. Topics to be covered: biotechnology, cell fusion, biological process, enzyme kinetics, industrial applications of enzymes, carbohydrates, plant cell cultivation. Biotechnology. The term biotechnology can be applied to the commercial techniques used by organizations or groups to make or modify a biological product. This includes the many methods used to improve the characteristics of economically important plants and animals or the development of valuable microorganisms. It is also used to cover new scientific procedures involving recombinant DNA and cell fusion. Recombinant DNA allows the direct manipulation of individual cells' genetic material, which may be used to develop microorganisms. This technique's primary objective is to splice a foreign gene into circular forms of DNA, plasmids, to produce the desired effect, which can then be incorporated into an organism. Cell fusion. Cell fusion is the combination of a single manufactured cell created from the nuclei and cytoplasm from two different types of material, merging two desirable characteristics into one. This has been fundamentally useful in the field of research and science and has enabled breakthroughs in the production of antibodies and medical treatments. For instance, hybridomas, hybrid cells, can be cultivated by combining specialized immunity system cells and tumor cells to produce antibodies for specific treatments, diagnoses, and purification. Recombinant DNA technology can also be used to develop genetically modified microorganisms to produce various chemical compounds with higher yields. The recombinant DNA or cell fusion technologies has enabled the scientific communities to develop exciting new and advantageous breeds of cells in astonishingly quick times. Along with the inherent potential of biotechnology, the successful commercialization of cell fusion procedures has led to large-scale operations that are continually researching this sector for economic and social possibilities. This is just one reason why biochemical engineering has become one of the most lucrative and significant areas in biotechnology and critical to its reputation and success. Biological process. Living cells or their components are used in biological processes to effect desired physical or chemical changes or enhancements. Biological processes have benefits and drawbacks over traditional chemical processes. The significant advantages include the following factors: mild reaction conditions. Bioprocesses have moderate reaction conditions. Room temperature, air pressure, and a relatively neutral medium pH are common conditions. As a result, the procedure is easier, and the production facilities are simpler than traditional chemical processes. Specificity enzyme catalysts are circumstantial and will only catalyze in a range of predictable chemical reactions. There are several enzymes that can catalyze a wide range of reactions. Effectiveness. The rate of an enzyme-catalyzed reaction is usually much faster than that of the same response when directed by other types of catalysts, and only a small amount of enzyme is required to produce the expected effect. Renewable resources. The significant raw material for bioprocesses is biomass, which is easily obtained and provides both the carbon skeletons and energy required for organic chemical manufacture synthesis. Recombinant DNA technology. The development of recombinant DNA technology promises enormous possibilities to improve biological processes. However, the application of biological processes can provide a number of disadvantages, which can include complex product mixtures. In cases of cell cultivation, microbial, animal, or plant, multiple enzyme reactions will occur during the process. This can lead to a final product that will contain a cell mass, many metabolic byproducts, and only a remnant of the original nutrients. Dilute aqueous environments. The commercial components are often only produced in small amounts in a water-based medium. Since products of bioprocesses are frequently sensitive to heat, traditional separation techniques cannot be employed. So, unique or customized techniques will have to be used, which can be expensive to scale up. Contamination. The fermenter system can easily be contaminated as environmental bacteria and mold can grow well on most moist surfaces. The problem becomes more difficult with plant or animal cells because their growth rates are much slower than those of environmental bacteria or molds. Variability. Cells can mutate because of the changing environment and may lose some vital characteristics needed to make the process successful. Enzymes are comparatively sensitive or unstable molecules that require care in their use.